We could test kidney medicine on this, or we could test it on this. We could test heart medicine on this, or we could test it on this. We could test lung medicine on this, or we could test it on these. According to Humane Society International, 115 million animals are used in lab experiments worldwide. But what if they didn't need to be? What if we didn't need real organs to test drugs? These are organs on chips. Today, I'm in Boston at Harvard's Wyss Institute for Biological Engineering. Since 1950, the number of approved drugs in the US has been decreasing, even though we're spending more money than ever on pharmaceuticals. And before drugs can be given to any actual human beings in a clinical trial, they have to go through a lengthy process of lab and animal testing. This is Don Ingber, a cell biologist and bioengineer. He's not the biggest fan of the drug development industry.
Right now they'll spend 500 million easily, or billions, 15 years, get to the clinical trial, have many thousands of patients, and fail. The drug development model is broken.
But Dr. Ingber likes a challenge. When Dr. Ingber was at Yale, he was studying molecular biology, but there was something else that caught his eye.
There were students walking around campus with structures that looked like jewels or geodesic dome-type structures, and I asked, what was that? And they said, oh, it's in an art class. I said, what's the name of the art class? And they said, three-dimensional design.
He talked his way into that art class by explaining to the professor that those sculptures reminded him of something.
I was learning that at the level of molecules, it's the shape of the molecule that dictates its function.
So on the first day of class, the professor hands out some wooden dowels and fishing line, and he tells the class to make an object out of them.
But the sticks can't touch. You could only pull them up into a 3D shape by strings, and it has to hold itself open.
The professor left. At first, nothing happened.
But all of a sudden, somebody who must have seen one of the sculptures of Kenneth Snelson, who built these structures called tensegrity, tensional integrity, built a 3-stick structure, and it's kind of wild. It's like you don't think it's possible.
The professor also had another model out, one that could collapse and pop back into shape, just like the molecules Dr. Ingber had been studying. So the next day, he went back to the biology department.
And we used a drug that caused the shape to change, and I said to the postdoc, the tensegrity changes.
So Dr. Ingber took what he learned in art class and applied it to his studies in the lab. Tensegrity became the basis of his career, and it led him to organs on chips. So here's an example of how a lung on a chip is used. Here in the middle, there's this tiny, tiny channel that can be filled with fluids. And at this small scale, fluids behave in some interesting ways. There's even a special name for it.
Microfluidics.
In these tiny channels, fluids don't mix with each other. They stay separated by a small membrane, but they can still interact with each other. Scientists fill one side with air and the other with a liquid that simulates blood. On the blood side, there are real living white blood cells. On the air side, there are human lung cells and bacteria. The air and the blood pass through the channel, and while that's happening, the sides expand and contract. This simulates breathing. And these are the kinds of forces Dr. Ingber was talking about. Turns out this is a pretty accurate representation of a lung, which is amazing. It means that cells in these tiny chips are interacting with each other like they do inside a real human. And this has some far-reaching benefits. This is Geraldine Hamilton. She's the president of Emulate, a company trying to use these chips to fix the broken pharmaceutical industry.
The current ways of testing if a drug is going to be safe or if it's going to work, if it's going to be efficacious, involve either cells in simple dishes or the use of animal testing.
That means subjecting animals to the dangers of untested drugs. Many of those tests don't even predict how the drugs will work in humans, which can lead to the drug failing in clinical trials.
The great thing about the organs on chips is that you could work on the mechanism of how it works on human, as opposed to work years on mouse and then find out it's different in human.
And that could help speed up the development of drugs and identify problems with them much earlier. This could be crucial in the case of an outbreak or an epidemic. On top of all of that, most drugs are developed to be one size fits all. That's not always the best approach. Organs on chips could help change that.
In the future, it could be your cells on the chip. I'm not just going to do a drug that treats 100,000 people, all of whom are potentially quite different genetically and their environments are different and so forth, but it's personalized to you.
Of course, clinical trials still require time and care to get it right. After all, making sure drugs are safe in humans is the whole reason the process exists. But if organs on chips can help speed up that process at all, or bypass putting animals at risk, they could be really beneficial. There are other groups in California, Germany, and Israel working on this kind of organ simulation too. But Emulate and the Wyss Institute may be uniquely suited to develop a mainstream solution. There's a vibrant blend of hard numbers and creativity, where scientists, engineers, and entrepreneurs are all excited about solving big problems.
At this institute, you have the right people, the right materials, some funding, and any crazy idea. If you can get other people excited, it just happens on its own. It's equally invigorating and exciting for me because I just love the creative process.
And it's that creative process, that entrepreneurial spirit, this mix of art and science, that could phase out animal testing and a whole lot more.
So good afternoon, everyone. We are the Group 5 and we're going to discuss to you microchip modeling clinical trials, or also known as organs in a chip. So at the moment, the methods we use to discover and develop new drugs is too expensive, it takes so much time, and it fails more than it succeeds. And nowadays, we seem to be spending more and more money in research and development, yet we are getting less drugs approved into the market. One of the key factors resulting to this circumstance is that the tools that we currently have to test is to test whether a drug is going to work, or whether it has efficacy, or whether it's going to be safe before we get into human clinical trials, are not providing accurate and precise results. So currently we have two main tools available before we can proceed to using human subjects in trials. These are cells in the dishes and animal testing. Animals do and can provide extremely useful information. They teach us about what happens inside a complex living organism. However, animal models fail to predict what will happen in humans when they're treated with a particular drug. Our bodies are dynamic environment, so if we want to make these cells thrive outside our bodies, there is a need to design, build, and engineer a home away from home for these cells. So in 2013, the Wyss Institute researchers at Harvard University and a multidisciplinary team of collaborators have engineered microchips that sum up the microarchitecture and functions of the living human organs. These microchips, called organ-on-chips, offer a potential alternative to traditional animal testing. So what is an organ on a chip? So these are living cells inside a little chip, cells that are in dynamic environment interacting with different cell types. So researchers are simply trying to recreate in this tiny chip the smallest functional unit that represents the biochemistry, the function, and the mechanical strain that the cells experience in our bodies. So how does it work? We use techniques from the computer chip manufacturing industry to make these structures at a scale relevant to both cells and the environment. We have three fluidic channels in the center. We have porous, flexible membrane on which we can add human cells from, say, our lungs, and then underneath, they had capillary cells, the cells in our blood vessels. And then we can apply mechanical forces on the chip that stretch and contract the membrane, so the cells experience the same mechanical forces that they did when we breathe. Lung on a chip. We could, for example, mimic infection, where we add bacterial cells into the lung, then we add human white blood cells. White blood cells are our body's defense against bacterial invaders. When these sense this inflammation due to infections, they will enter the blood cells into the lung and engulf the bacteria.
So other example is a gut on a chip. So inside this chip has an intestinal human cell in a gut. As you see in the left picture, dyan nilalagay yung intestinal cell. And in the right photo, this is how it looks like inside that chip. It can mimic many of the functions that you would actually expect to see in a human intestine. And with that, pwede ka na mag-create ng mga models of diseases such as irritable bowel syndrome. So we see in here that it can create a better comprehension inside human body. The bottom of the picture is a closer look on a chip. So as you see in the upper picture, it is a human with a lung, a heart, bone marrow, and etc. Now, the true power of this technology really comes from the fact that it can fluidically link them. This is not going to ever recreate a whole human in these chips, but the goal is to create sufficient functionality so that it can make better prediction of what is going to happen in humans. So in this one, this is how our body responds to a certain drug. So for example, exploring what happens when we put an aerosol drug. So if you have an asthma, when you take an inhaler, so using this chip, we can fully understand how it enters our body. Does it have toxicity? Is it metabolized in the liver? Is it excreted in the kidneys? We can begin to study the dynamic response of the body to a drug. So, possible applications. So, we have skin on a chip. Basically, nilalagay ang human skin in a chip and then magagawa niyang gayahin yung mga normal function ng isang skin sa human. This is a test whether the ingredients in those products that you are using are safe to put in your skin without the need of animal testing. So the other one is we could test the safety of the chemicals that we are exposed to our daily basis in our environment, such as chemicals in regular household cleaners. So we could also use the organs on chip for application in biotourism and radiation exposure. Clinical trials on chips. So right now, the average participant in a clinical trial tends to be middle-aged, tends to be female. You won't find many clinical trials in which children are involved. Yet every day we give children medications. Childrens are not adults. Now imagine if it could take cells from all of those different populations, put them on chips, and create population on a chip. This may save countless lives of animals that are used in clinical trials.
Next is team. So a team is a group of people working together to achieve a certain goal, and the team working together in WIS Institute consists of engineers, cell biologists, clinicians, and more. The individuals involved are and should be influenced by biology. Next is instrument. The instrument is going to give the manufacturers the capability to link 10 or more organ chips together. Basically, one can just take a chip, put it in a cartridge, as you can see in this illustration, the blue one, and then put the cartridge into the machine. So what a future might look like if I could take your stem cells and put them on a chip. Basically, it would be a personalized chip just for you. Now, all of us are unique individuals, and those individual differences mean that we can react very differently and sometimes in unpredictable ways to drugs. This would sometimes lead to adverse reactions and it could be fatal. So, how do we prevent this? We could imagine one day you on a chip, and this chip would show the precise reaction of your body or cells to a drug. New developments. First off, we have for COVID-19. As organs on chips predict human responses with great precision, it can be used to better understand COVID-19 since they mimic the organ functions, giving us a more precise result for human use. Recently, alveolus lung chip and an airway lung chip have been developed, and since these chips are relevant for COVID-19 research would accelerate. Next is human on a chip. Human on a chip is a recent development in which multiple organ chips are linked together. This would allow the scientists to assess the drug's pharmacokinetics and pharmacodynamics across the entire link system. 8 different organ chips are involved. It includes the intestine, liver, kidney, heart, lung, skin, blood-brain barrier, and brain. So next is personalized medicine. Organ-on-a-chip technologies may accelerate the journey to personalized medicine. Growing a patient's cells using an organ-on-a-chip could be a way to find out the unique response of his or her cells to a drug. The following is a summary of its benefits. So it provides an accurate alternative to traditional animal tests, tests the safety of cosmetics and also its toxicity, and helps scientists elucidate how tissues respond to a new drug candidate. So lastly, organ-on-a-chip improves the lack of predictive value. So we have this current lack of precision in medicine that contributes to inefficient healthcare and may not beneficial to the patient. But with organ-on-a-chip, it predicts human response precisely in which it may be the solution to the lengthy and time-consuming drug development.